pleasure to introduce Dr. Matthew Gonnerman. Matt is in my lab in conjunction with Dr. Diane Prosser as well at USGS. And I have consistently had a postdoc that kind of helps guide a bunch of my graduate students. And I had an opening for an avian influenza position in 2022, which I think we were interviewing in 2021. But I wanted something a little bit more unique. I didn't want somebody, a traditional postdoc that just came in and did their own work. I wanted somebody who's actually going to work with my students, because it was good advice that had been given to me to have somebody who also helps mentor students. And so when I was interviewing people, that's what I was looking for, somebody who was driven and quantitative, but also interested in guiding students. And I was very lucky to end up being able to hire Dr. Gonnerman, and that is the role he plays. So he comes to us from a long history of trapping all kinds of animals, from quail to turkeys to all kinds of things you're going to hear about. He did his master's at Auburn, and then he did his PhD at the University of Maine, both of which are on the subject of wild turkeys, and his dissertation in particular was kind of disease ecology based. In my lab, he's working exclusively, his own science is working on high-path avian influenza. And he's going to present a lot of that work today, along with giving you some of his background, looking at multiple different publications he's worked on. But keep in mind that he has lots of different tools, and so he's helping lots of different of my students on their projects as well. And so I'm super appreciative of him being here. He's a fabulous scientist.
Thank you very much, Jennifer. That was very, very kind. Jennifer just kind of gave you a little bit of my background. I'll give you a little bit more. So my master's and my PhD were both with wild turkeys, and I spent a lot of that time in the field trapping. But when I was done trapping and collecting all the data, I also spent a lot of time behind a computer modeling. So a good portion of my experience is in population and movement modeling of wild animals, and especially in that in the context of disease dynamics and understanding how they impact those aspects of animals' life. While there's plenty of stuff to read, if you would rather just watch a video about my research, there is a National Wild Turkey Federation did a video called Colonial Forests, Are There Too Many Turkeys in Maine? There's everything from field work all the way through the epidemiology to check out there. But today, we're going to talk about a lighter subject. We're going to talk about the spillover of avian influenza into our agricultural systems. Generally, avian influenza viruses can also just be referred to as influenza A viruses. These are a larger group of viruses that impact both birds and mammals. We generally specify them according to their surface proteins. So when you hear H5N1, H5H7, those are referring to the different neuraminidase and hemagglutinin surface proteins on a given virus. More broadly, we can also organize our viruses into low and high pathogenicity. So how dramatically, how much morbidity and mortality do we experience in domestic poultry specifically? So we humans have a large poultry industry, and the avian influenza impacts us through that most directly. And so we have tended to group our viruses according to that nomenclature. Historically, low pathogenic avian influenza has been the primary dominant strain of avian influenza circulating in wild birds. These circulate locally amongst waterfowl and other water birds, but then can also be moved long distances by migratory birds of any type. There is a high rate of reassortment and mutations among avian influenza, which makes it a very dynamic disease system. When I say reassortments, that means that two independent viruses infect the same individual, and their DNA essentially is mixed together, and you produce a new viral outcome, a new subtype from those two viruses. And then mutations, obviously, one virus mutates into a new strain. And then as far as transmission, we are very focused on the viral shedding within animals. So animals can excrete the virus either through the respiratory system, through their gastrointestinal system. So virus in birds generally is transmitted via the respiratory and fecal routes. However, there's also virus in the actual tissue of the animal, and the organs of the animal. So essentially a highly infected, or an animal that is shedding a lot of virus could be shedding it through many different routes. While we will talk about a number of different hosts today, the primary host that I'll be focusing on is waterfowl as a group. So avian influenza viruses persist in wetter and cooler environments in general, and this tends to overlap with our freshwater and aquatic foragers. We talk about them broadly as including ducks, goose, and swan, but one of the subgroups of this, the dabbling duck, is a very highly impacted group of waterfowl because of the way that they forage. So a dabbling duck essentially upends itself, sticks its head in the water, and dabbles at the surface of the surface of the water source that it's foraging in. At the same time, an infected animal might be swimming by and excreting virus via its feces, and it is in the substrate that the animal is foraging in, so there is a direct contact with that infectious material. And so it tends to have a higher rate of infection across these animals. We also have to take into account that these animals can migrate. So waterfowl migrate north-south along multiple flyways, with some mixing across those flyways. So there's both compartmentalized movement up and down the United States and the Americas, but also some mixing across those groups. And within those individual flyways, we have variable but generally high densities of waterfowl depending on what species we're talking about. And those densities tend to overlap with agriculture, as these species prefer habitat that is associated with those land uses. And this leads to an important term that I'll use a lot today, spillovers. A spillover is when an infection goes from one system into another system, usually from wild into agriculture. I might use the term spillback. This is if agriculture then has an infection that goes back into the wild system. So why would we care at all about a bird flu? Well, as many of you probably are already aware, the food security issues related to avian influenza are pretty dramatic here across the world because of the impacts to the poultry and more recently to the cattle industry. So whenever an infection is identified at a poultry farm, there is a necessity to essentially cull the entire population. That can lead to an immense amount of economic loss for that industry. Similarly, if there is an infection at a dairy farm, while pasteurization does neutralize the virus, out of an abundance of caution in the early days of the outbreak, there was a bunch of milk dumping, which was also an economic loss. And then finally, while it hasn't happened this outbreak, swine is an important industry to keep an eye on, both because pork is a key food source for many Americans, but also because swine are able to contract both avian forms and human forms of influenza, which means that they are a perfect animal for reassortment that could lead to a zoonotic disease. When I say zoonotic disease, I mean a wildlife disease that infects and circulates among humans. And that is one of the primary fears for avian influenza. We already have infections in poultry and dairy farm workers, and luckily there's currently no evidence of human-to-human transfer among those workers, but that would be the final step towards a zoonotic outbreak. And while maybe lesser for some people, we have a pretty broad need to understand this in terms of wildlife conservation. We have a number of at-risk bird species across the United States, which could be impacted dramatically. This is most evident in mass mortality events, which we'll talk about a little bit later. So while there are a ton of different types of avian influenzas, I'm going to focus on the H5N1 highly pathogenic strain and the subsequent system that it created in the United States. So in late 2021, H5N1 was introduced across the Atlantic from Europe into North America. This in and of itself was a rather rare occurrence, as the tendency is for influenza to come across the Pacific. Once it entered into the United States, there was a dramatic increase in the number of wild bird and domestic poultry outbreaks that we observed across the nation, with specific hot spots occurring in different parts of the nation. But after a time, we saw that these outbreaks became more seasonal in nature, and although they were a bit variable in magnitude depending on the year. And this seasonal nature gives the impression of the potential for endemicity. So the virus, when it introduced, had variable, unpredictable dynamics, but as it settles into an endemic state with the waterfowl seasonal movements, we may expect this to become more predictable if it becomes endemic. And after, in 2023, at the end of 2023, we started to experience a number of spillover into dairy with infections in mammals, and I'll save a little bit more of that for later as well. So as you can see, there is a huge amount of complexity and ambiguity in this wild bird agricultural system. We'll call it the wild bird agricultural interface. So where does that wild bird and poultry or dairy system interact? There's a ton of ambiguity in host roles. So while I said that waterfowl are the primary host for avian influenza in the wild, there's not a whole lot of understanding of what is happening in this current H5N1 dynamics system. So what are the species that are moving the virus long distances? Who is circulating it locally? And especially who's transporting that virus onto the farms? Because it's very unclear how the virus is currently entering the farms, partly because there's so many different pathways and hosts that could be moving it. So up here, this image here is just to kind of give you an idea of the variability in host dynamics. So we consider hosts in terms of their infection rate, their mortality, their viral shedding. There's each of these individual hosts can differ in each of those specific traits and impact their ability to move the virus. Similarly, within a individual poultry system, there are a number of different entryways, some related to wild birds, some completely unrelated to wild birds, that could be driving transmission into a poultry farm. So rodents, small birds, waterfowl on local water holes, or trucks and employees, all potential entry points for the virus. On top of the ambiguity on the individual transmission dynamics, the broader prevalence and risk dynamics across the landscape are constantly in flux because of weather and seasonal movements of animals. So again, the virus is only viable in certain conditions, and it requires hosts to be around for there to be infections in the first place. So as those dynamics shift, we'll see changes in the dominant strain, changes in the number of hosts in an area, and it pretty much creates an immense amount of uncertainty in understanding a given system. So to understand that uncertainty, the USGS and the USDA have collaborated to try and make sense of what information we have available to us in describing risk of transmission to both wild and agricultural systems. So specifically, where and when are the highest risk areas for AIV spread between wild birds and poultry in the United States? To answer this, we have a number of different data sources available to us that have been organized through efforts by the USDA and the USGS. So most prominently, we have live bird surveillance that occurs across the United States. This is driven by a quota system where essentially we set quotas for given regions, and then counties go out, sample animals, swab them, and then test them for active avian influenza infections. And that information can give us a time and location for when and if avian influenza was on the landscape. As I mentioned, waterfowl are our primary host species, and we also happen to harvest a large number of waterfowl across the United States. And so that information can also be used to look for and identify avian influenza infections. And finally, within the wild system, we can also use mortality events as some spatial temporal indicator of infections. However, these tend to be biased towards mass mortalities as they're much more easy to encounter as opposed to an individual animal on the landscape. And then as far as the domestic system, the information coming out of cattle farms and poultry farms is largely driven by self-testing and reporting, but there is a large amount of incentive to work with the USDA to be a part of these programs that work on biosecurity and protect an individual farm. So it's a bit of everyone helps each other by surveilling themselves. So before I came here, my predecessor postdoc Cody Kent published a paper discussing the early efforts to describe this system in terms of the prevalence of AIV in wild waterfowl. Now, Cody was working on this model before the I believe it was actually during the outbreak. So none of the actual avian H5N1 highly pathogenic information was available at the time. So Cody built a prevalence models based off of low pathogenic and a small number of outbreaks that occurred in 2015. And the impetus for this project was to, one, provide a spatiotemporal estimate of prevalence across the United States for a given species, but also to identify the differences among species in how they react to the virus. So zooming in on a couple of specific species, we can see that for the exact same time frame, we have relatively similarly timed increases in prevalence, but differences in magnitude. So we can use this information to describe risk in other ways. So this model was then taken, these estimates of when and where on the landscape ducks were infected with, or expected that ducks would be infected with the virus. And we took that and combined that with information on poultry systems to create a mechanistic model for risk. So in this model, risk is described as the distribution of waterfowl overlapping with the distribution of poultry, but we weight each according to their relationship to the virus. So we weight waterfowl according to the prevalence within a given species, so that's Cody's prevalence model. And then we weight poultry distributions according to the expected susceptibility of a poultry operation based on its production type. So, for example, we expect that commercial farms might have a greater risk than backyard farms, depending on their intake. We have some expectations that different commodity types, so broilers versus layers, may have different risk types. And all of those, the weighted differences in risk across these different production types were applied to the distributions uniformly to try and give a better idea of what risk is like on the landscape. And this is that model, this mechanistic model. It was validated against real outbreak data that was occurring in 2022. So the mechanistic model is not based on any actual H5N1 data. However, just using the movements of waterfowl and the distribution of poultry, we are able to pretty consistently predict high-risk areas across the nation. However, as I said, the susceptibility component of this model is relatively simplistic, and there may be better ways to describe susceptibility at a given poultry farm. For one, we based this information off of studies in Europe, which has a different agricultural system than us, so there was some expectation that it would just inherently be different. But we also do not expect risk to be uniform in space and time as it was assumed in that mechanistic model. So we wanted to move from a mechanistic expectation-based approach to a more empirical data-driven approach. And to do that, we took that validation data. So we had that 2022 spillover information that was describing when and where an outbreak occurred on a poultry farm. And we used that as inputs for a basic regression model where we were trying to describe risk according to specific land use types, the climate at a given area, as well as the specific production in poultry that was, the production type in poultry that was on the, was at a specific facility. On top of all of that, within the framework that we applied, we were able to account specifically for the spatial temporal dynamics. So explicitly taking out the time, we can essentially map the entire United States and show where we have higher than expected risk. At the same time, we had access to a case control questionnaire data set, which surveyed commercial turkey farms during that first year of the outbreak. So we have a commercial turkey farm, it has an outbreak on it. This survey is sent to them and asks them specific questions about biosecurity that was taking place during the outbreak. We then send a second survey to a control farm in the vicinity of that outbreak farm. So we have, and we asked them those same questions. So we have information on what biosecurity was taking place at a successful farm that did not have an outbreak and one that did have an outbreak. And then we can integrate that information with the larger spatial temporal data set to describe environmental risk and risk associated with individual biosecurity metrics, accounting for the landscape that that farm is existing in. Jumping into results, we saw certain things that were completely intuitive. For example, our temporal relationships were consistent with the migratory season of waterfowl, which we would have expected. We also saw spatial temporal relationships that were consistent with where we saw outbreaks occurring, which makes sense. But just to give you an idea, here on the left, we have the cumulative risk across the United States. And then here on the right, annual cumulative risk. And then here on the right, we have broken up into 6-week increments. So you can see that while there were consistent areas of high risk, it is dynamic across the landscape, and your risk is not necessarily always going to be high throughout the year if you are in a high risk area. When diving deeper into the environmental correlates, we found that relative waterfowl abundances within the previous 2 weeks were indicative of risk, as well as the decrease in mean temperature, as well as an increase in cumulative precipitation. So cooler, wetter environments, which again, consistent with our understanding of the virus. We also found some interesting dynamics associated with human land use, as well as certain aspects associated with waterfowl distributions. So human population density and proportion developed were both associated with higher risk. And we saw that proportion of flowing water, decrease in proportion of flowing water, as well as an increase in NDVI. So NDVI essentially is tracking the amount of green vegetation on the landscape. So when you have an increase in green up, that is also, you're seeing a slight increase in risk. Diving into the biosecurity measures that were impactful for reducing risk, we actually identified quite a few different practices that can be impactful for reducing risk. One of the more jarring relationships that we found was that as you increase the number of vehicles on a given facility, your risk increases to nearly 100% after a certain point. So after you have 40 or so vehicles entering your site, you have a very high chance of a spillover event. However, if you are using a wash station, that risk is dramatically reduced. Obviously, you can see that there's some uncertainty at the farther end of these bounds, but there is a clear signal in this data set that wash stations are impactful for reducing risk, and that there is some mechanism by which the trucks may be moving virus onto these facilities. We also saw a number of other actions that farmers could take that are relatively prevalent in the industry. So, for example, changing how you use where you're disposing of dead birds, so not sharing it with another facility is very important. Treating your litter, so when you have these poultry farms, there's essentially litter on the ground. You need to change that every once in a while. When they do that, they can heat treat it, they can bring in fresh litter, or they can sometimes just bring out old litter, heat it, and bring it back in. So there's different ways in which they will adjust the litter that can impact the risk. And then another finding that I found interesting was that a restroom in the facility also reduces risk. So you have a situation where you have employees who maybe have to use the restroom, but have to walk across the entire farm or walk out into the field or behind the farm into a place where there is potentially virus that they could pick up and then bring back into the facility. It's better to just have a restroom in the facility so that people aren't walking around. All of this has recently been published and is going to be in a special issue in the journal One Health at the end of the year, so I encourage you all to check that out. But I'm going to switch gears a little bit here. So we've been talking a lot about waterfowl, but it's important to note that waterfowl are not the only host of highly pathogenic avian influenza. We tend to see species that are gregarious be more of the interesting hosts that we look into. So group feeders, colonial breeders, anything where you're spending or you have a high density of animals on top of each other, and especially aquatic foragers. But one that maybe we haven't talked about too much yet is predators and scavengers. So birds are infected with the virus, they have virus in their organs and their muscles, and then a raptor comes through and kills that bird and then eats it, and now it is highly at risk of getting infected. So these animals that eat infected animals are themselves at risk. So within all of the hosts, we tend to group them according to how well they move the virus and how susceptible they are to the virus. So for the potential spreaders, we focus on the waterfowl, the dabbling ducks, as well as gulls. So gulls have a high movement capacity and they have a high infection rate. So they have a capacity to get infected, move a long distance, and move the virus across the United States. Within our at-risk bird communities, we tend to focus on water birds, seabirds, as they are more frequently group feeders and colonial breeders. We're also looking into carrion eaters and raptors. So there's been a number of vulture outbreaks in the area related to potentially scavenging on dead infected animals. One ambiguous group is the passerines. So passerines themselves are low infection rate, low shed rate animals, but they exist in high densities. So you can have a small number of animals that are actually, or excuse me, you can have a small proportion of a population that is actually infected, but a large number of animals. And they additionally have a lot of ability to access areas that they shouldn't, like on farms. So there's some potential that these could be animals that are moving the virus around. One way to differentiate hosts is by looking at how they respond to the virus, but that is very difficult to do in wild settings. So we use challenge studies where we capture animals, we inoculate them with the virus through whatever method we're currently testing, and we assess their response. We assess the rate of infections, the rate of mortalities in the animals that we tested, and we assess the rate and the magnitude and the duration of the shedding of the virus. So we can compare these. This data is a review of essentially every challenge study that I could find in the literature and summarized for each species that we were interested in. And then once we have that information, we can then look across species to compare them. So on the top here, you'll see infection rates as X's and mortality rates as red dots. Where you have a greater distance between infection and mortality, that is an animal that is has a greater capacity for infection with a lesser capacity for mortality. So that would be an animal that can take up the virus but isn't going to be killed by it. Similarly, the farther you are to the right on either of these two bottom charts indicates your ability to shed the virus. So if we look across these traits, we can see that wood duck has a higher mortality rate and higher shedding rates, which would make it a moderately good spreader of the disease, whereas mallards have low mortality rates and moderate to high infection rates, moderate to high shedding rates, which would indicate that they're probably a better spreader of the disease. Unfortunately, disease response is only one component of the host's ability to spread the virus. We also must take into account their life history strategies as well as their ecological niche. So Jennifer's lab and Dee's lab kind of came together, led by myself and another postdoc, and we tried to formulate a means to describe the host system. And we've essentially come up with this proposed continuum where we can plot animals according to their disease response traits and their life history strategies, and describe them in how vulnerable they are to HPI, and how competent they are at spreading the disease. I'm not going to get into the details of all of this because it's still in revisions, but broadly, what we are doing is taking data sets that are publicly available. So these are, we did not collect this information. We took my challenge study review. We collected a couple of other life history data. We took some information from life history databases, and we combined that all into several different non-parametric qualitative analyses, which allowed us to group and rank these different bird hosts according to our expectations for a given trait. So we set our hypotheses: higher infection rate, you are a more competent host, lower mortality rate, higher competency, and so on. You can put all of those individual hypotheses and data on traits into our analyses and come out with essentially a two-dimensional axes describing the host system. And then we can take the host system and look at the overlap in species, look at their ecological niche. What are the likely interactions between highly susceptible and highly competent species? I would love to dive into the interesting interactions between the different species, but one of the major findings we have from this exercise is that we don't really have the data to fully describe the avian influenza disease system for wild birds. And this is largely because of a hyper-focus on Anatidae, which is the waterfowl species, as a primary host, and a lack of studies on other species. So here on the right, I have essentially quantified the number of challenge studies against the number of mortalities in the USDA database. And you'll see that while the waterfowl are pretty well represented, we have a large chunk of animals that are dying in the USDA database that are not also being challenged by our experimental studies. This includes pelicans, vultures, a huge number of different diverse species are represented here. And this represents areas of need for research. And moving beyond just birds, we are also now having to expand our expectations of risk into mammals and especially the dairy system. So again, we've talked about reassortments allowing the virus to change and impact new species. Well, that allows it also to jump into mammals and eventually us potentially. We've already seen a number of different jumps from bird to mammals on these poultry and dairy farms. So oftentimes when we have these outbreaks, they'll go and they'll just sample everything on these farms to check and see what is infected. And at these outbreaks, we found small mammals have been infected, pets, employees on mixed use farms. We found all of the animals essentially on that farm have been infected in some cases, all of the types of animals have been infected. And these different species being impacted also lend themselves to new transmission pathways. So for example, rodents being infected allows for a new pathway into and out of farms or into homes. Rodents have an amazing capacity to get into wherever they want. Similarly, those rodents could be eaten by cats, or the cat could be eating infected birds, and that pet can then take it into your house, or it might die itself. There's been a number of mortalities in domestic cats on these poultry farms. But the biggest issue right now in terms of mammal infections is the dairy cattle system. So in late 2023, we saw the first of what has turned into upwards of 1,000 dairy cattle farms infected by avian influenza. A key thing to note here is that only 3 of these were actually spillover events. So a large component of this is farm-to-farm transfer between what we believe between dairy cattle farms. And this has led to a high amount of spatial clustering on the landscape. So over here on the left, you can see our cattle outbreaks in red and our poultry outbreaks in blue. You'll see that it's not easy to tell here, but there's actually twice as many dairy outbreaks as poultry outbreaks because they're so clustered on top of each other. And with that high spatial clustering, does that increase the potential for transfer to poultry or wild bird systems? And so I'll leave you with a little bit of what I'm working on now. So my current research that I'm focusing on is assessing the transmission dynamics associated with these dairy cattle systems. So we have a outbreak that occurs on a farm, and we know that some amount of farms in the area are also going to be impacted via farm-to-farm spread, but some other dairy cattle farms. What we don't know is if that dairy cattle farm is also infecting the local environment via some unknown mechanism, or if there is also continued farm-to-farm spread to poultry systems. While we just kind of started on this research, but there's a high amount of overlap between specific B313 viral subtypes in poultry systems and cattle systems. And so that linkage, understanding the viral phenotype, allows us to look at specific combinations and maybe even understand the network of transmission that's happening. I know that was a lot, so I'm going to end there and leave time for questions. I also just really want to note that this is a hugely collaborative effort led by Jennifer Molinax and Dee Prosser, and at least 4 other postdocs have worked on this. And so there's just a lot of people working on this system right now that I want to make sure that they get their due recognition.
So 2 questions. One, how many, for it to be considered an outbreak, is it just one bird having the disease? And if a farmer has an outbreak, what do they do like afterwards?
So an outbreak would not be considered one animal, but also it's unlikely to be one animal that you would even detect it. Usually what's happening is, I believe there's some amount of testing, but also a chicken that is impacted by avian influenza has very obvious neurological symptoms in a highly pathogenic case. So generally what will happen is you'll start seeing that it'll visually happen, and then that will be an indicator that sampling needs to occur. And then again, it depends on the facility and the production size. Higher production output will have a higher probably biosecurity standard and a higher or a larger amount of resources to place towards surveillance and things like that. So it's really dependent on the system as far as testing. But then regardless, that if you do identify an outbreak, you do then communicate with USDA. And then they're keeping track of that and working with people. The birds are culled. If you have an outbreak on your farm, there's specific guidances, but essentially at the very least you're killing every bird in that building, usually.
Hi. How are farming systems unique between Europe and North America? That's the thing you touched on in your piece about the model.
Yeah, so that's a great question. I would say that that's not my area of expertise. My understanding is that the production level in the United States is much higher. We have denser farms, and I believe that there's very specific minute differences in just like the materials that you use for your substrate and things like that that can vary between the systems, but at a broader scale,
It's size.
yeah, it's size.
Okay.
Great talk, Matt.
Oh, thanks.
I know you're just working on this new research question, and I know we gossiped about it getting coffee recently.
Mm-hmm.
What management do you think, outside of, you said wash stations are good, something that's happening with trucks, maybe washing the trucks, for the cattle farms, because that's pretty shocking. And
Yeah.
is there more management that they can do or different for cattle farms?
There's some conversations that we need to have. So we've been so inundated with the poultry system that we don't have a whole, like, we as the lab don't have a whole lot of knowledge of the dairy system necessarily. So we need to contact some of these people. One thing that I'm thinking is that they need to be washing their farms when they're leaving the facility as opposed to just when they're entering. So if the virus is somehow just getting transported by the trucks, if it's not being picked up on the roads, which is less likely than them just picking it up at their facility where the outbreak's happening. So I think my easy one would be the wash station at leaving as well as entering. I don't know if that's necessarily common.
The dairy just don't have that security.
That's the other problem.
And most of these are milk farms, so you have milk trucks coming and going regularly where you don't have that for poultry farms. So the whole paradigm is gonna have to be revisited. You also have cows coming in and out. They don't typically stay in the barn. So the whole system is different, so they're going to have to approach it from a fresh start of how do we create biosecurity.
Can I ask like another maybe scary question? Is it in the milk? Like, is that one?
So that was actually, I was just gonna say, so yes, it's in the milk. It's primarily in the milk. It is neutralized by pasteurization. I don't know that they pasteurized the milk that they were dumping in to the environment. So that is like a key question, right? Like if it is in the milk and then they just dump a bunch of milk out into a lake or whatever, their surface water, and then waterfowl are there, that is a direct spillback event potentially. We don't know that happened, and we don't have genotyping to say one way or the other. So essentially right now, my hope is that I can take this spatial information and try and elucidate some of these relationships and then drive further research so that we can encourage people to go out and actually sample and do some of the genotyping of these wild birds and compare them to the domestic systems.
So you mentioned the dabbling ducks at the beginning and the substrates of the lakes, for instance, that they get into. And I would think how, yeah, you also mentioned how the viruses can, new viruses can be created, two viruses make selective generation. So how quickly is the progress, or how quickly can the virus change? And then,
Very quick, like I mean,
in like one generation.
yeah, so essentially, if that reassortment happens, that is a new virus. So like it's instantaneous, really. Like it essentially takes one co-infection, right? So you need to have two strains circulating in the same system first off, or two interfaces with a lot of infections. So if you have a human with an influenza virus working on a poultry farm, or maybe the better thing is like a person on a swine farm, and then a passerine comes in, they both infect that pig. That is now a highly pathogenic avian influenza that could infect humans.
and then so it'd be difficult to track certain like types of virus to a certain species of bird, for instance, because it's so spread.
Well, so it depends. So with the phylogenetics, we can if we have sampling in the species, it's possible. So essentially, what we've been able to do is within the challenge studies, for example, they try and grab specific viruses from specific animals. So we are sampling a lot of different viruses across the landscape. We keep track of those subtypes. There's some capacity if you do the actual genotyping to look at their relationships. The problem is actually doing that, the resources and the cost of getting that done, and also the sampling and which samples do you combine. So, for example, we have the farm-to-farm information on cattle, but they didn't include poultry in that, so we don't know if poultry is related to that system at all. We would have to rerun that entire analysis with the poultry included to understand. So the phylogenetics get a little bit more complicated and kind of dependent on the sampling that you're using.
Thank you for the talk and your work. Are you able to speak to the virus's persistence in the environment, like different environmental systems, whether it's aquatic or soil, just thinking about wild animal movement through infected patches?
Yeah, absolutely. So generally speaking, we're talking on the time scale of days to maybe a couple weeks. However, there are instances where it can persist up to months in specific environments. So that especially leads to a number of transmission issues. Specifically, you could have an entire population completely be immune, or like have everyone be immune, and then you enter in an area and now you're suddenly having a giant outbreak, even though there were no infected animals, except that they were there months prior. So
poultry houses have these high volume fans because of ammonia. So if people looked at directional studies, like dry deposition and airborne virus loads.
We've talked about it as a group, and we've discussed the potential for essentially the airflow. So they put windbreaks up on farms and things like that. We've discussed the existence of windbreaks to potentially disrupt the spread of the virus, but I don't think we've discussed specifically the fans on these facilities.
Yeah, so they, in China, they're doing bioaerosol collection inside of poultry houses, and they figured it out. And School of Public Health right now is putting a proposal together to start to try and do this here. It's been done less in the US, where we're actually collecting air on either side of the fan. They've done sampling right on the fans. It's a point of interest with USDA right now, mainly because there's a lot of, we don't know how. Like with some of these farms that are so biosecure tight, it makes no sense how it's getting in. It's a good question.
That's an academic question. You mentioned the mechanistic modeling. So is that diffusion based? Is that a partial physical based, like differential equations that can give you spatial and temporal distribution of the virus?
So the mechanistic model that was implemented in the paper that I showed here is actually relatively simplistic. We are literally just doing arithmetic. We're taking the So the waterfowl distribution here is a spatial layer produced by eBird. They produce what's called eBird status and trends. It's the expected density of a given species at a given location. We take that and combine it with Cody's prevalence data, and we just essentially keep multiplying on top. We take the distribution of poultry times this uniform table here, and then we just multiply those two together. So it's a spatial temporal estimate, but there's no advanced like statistics going on or anything like that.
And that's a regression-based
Yeah.
model. So if the
Well,
models already exist, that then are then
Yeah.
applied together.
So along those lines, what opportunities do you see in using artificial intelligence, specifically like the newest versions of ChatGPT, to help you develop models and to look for more of a systems perspective on all the things that are going on?
I would say
Tell
that
what you really think, Matt.
I would say that I've not thought about it. I am not super integrated into the AI stuff, but what I would say is that the amount of information we're trying to deal with, there is some capacity, I think, for AI to kind of synthesize and try and identify key patterns. Because that's usually what it's good for, in my opinion. It's like this is a very complex and nuanced system. That's not generally something I would give to an AI to deal with and try and figure out. So yeah, I don't have a good answer for
I'm
that.
saying like the other perspective is you back off and you think about all the systems ecology perspective. You have all these stocks and flows and mechanisms, the virus is mutating. Having a global understanding of all that, I think AI can really do a nice job of helping you with that and give it the right kinds of models.
Yeah, like so.
It's not a data intense, it's a process to help you understand where to be looking.
Mhm.
Focus your efforts.
There was a PhD presentation on the Canadian geese migration that was based on, again, physical-based, more diffusion, differential equation-based equations. With Montas, one of the previous faculty members in bioengineering, did it. Mr. Goodlaw did that, one of the PhD students, the dissertation. So I think that this project, if you ask ChatGPT, probably rather than regression multiplications, it would probably suggest to you some diffusion type differential equation that can really give you a both time and spatial distribution of the viruses.
If we had the level of data you would need for a diffusion model.
Yeah, I mean, so like I said, there's some amount of limitations by the data that we're taking in. So we have to sample across the United States irregularly, and that sampling effort is not even. And so, for example, while you see a completely filled in map here, we're probably only sampling in 1 out of every 5 counties or something like that, and using the regression model to fill in the rest. Yeah, I have to look into that. I'm not familiar with that.
So I think we might have to leave it at that all. So
Thank you.
thanks
Thank
again.
you.